Hello and welcome back. My name is Marcel, and in this video we're going to solve the problem about the reverse complement of a DNA strand. Okay, so here is our problem in our Rosalind website. So here is the explanation about DNA. Okay, and the problem will be that in DNA strings we have symbols about that we use to refer of our nucleotides. Okay, so we have A and T that are complement of each other, and we have C and G which are complement to each other. Okay, so in this problem we're going to receive a DNA string of length at most 1,000 nucleotides, and we have to generate the reverse complement. So what is the reverse complement of DNA string? Okay, so I will use this example here to show you. Okay, so this is a DNA strand. Okay, and we should remember that every DNA strand has what we call polarity. Okay, and we define polarity from the C prime to the 3 prime. Okay, that's good. So the complementary string of this string will be T T T T G G G C C and A. Okay, the letters doesn't fit perfectly, but let's agree that's the complementary string. Okay, so this is complementary string, but the polarity of the complementary strand, it will be reversed. Okay, so this is 5 prime 3 prime. Okay, so as we should remember, every DNA strand we represent in the 5 prime 3 prime way. Okay, so what we have to do here is to reverse the complementary strand. So we have A, C, C, G, 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 and T, T, T. Okay, and then finally we have the reverse complement. Okay, let's put it the red way to be. Okay, let's check if is the output that they expect. Great, that's the output that the software expect. Okay, so let's move on to the VS Code so we can model our problem. So here in VS Code, what I'm gonna do is to create a function called reverse complement that will have an argument called DNA string, which should be a string and should return a string. Okay, the first thing that I will do here is to copy this test. Okay, so this way we'll be able to check if the string that the user input in this function are working well. Okay, so now I'm going to define a complement map. Okay, the complement map, which will be a dictionary. So A with T, T with A, then C with G, and G with C. Okay, my bad here. Now it's okay. So we define a complement map. So now let's move to the return and we're gonna do an empty string, then we're gonna use the join method. Okay, so now what I'm gonna do is to use the complement map get, and I will use base. Okay, so for each base in reversed DNA string, for each base in DNA string, we're gonna get the complementary base. Okay, looks nice, right? So let's test it. So I will just copy it here. I'm gonna be really fast. Our sample data set. Okay. Oops. Yeah, we're back. So let's define our sequence here. Then I'm going to apply the reverse complement of this string. What I have done here. Okay. So now let's run it and check if it works. It's important that to run our whole script because we have here all the functions defined. So they run directly on terminal. So let's try it this way. So define the first function. To be honest, I only need the validate DNA string. So let's move here. I'll define our sequence and print our reverse complement. So looks nice, right? As we can see here, this is the right answer. Okay, so now it's time that we move to write the code to solve this function, to solve this problem. Okay, so I use import sys, import os. So now from source tools, DNA tools, I'm going to import reverse complement. Okay, so I'm going to append the path to our file. Okay, and I will define the solve function, which has no argument, and then I'm gonna use with open. Okay, and here we're gonna use an R string. So this will probably data. We're gonna copy later. Okay, let's just keep it this way, and we're gonna use in the read mode. So this is F. We're gonna define the DNA seq as f.read.strip. We're going to strip the lines. Then I'm going to define the reverse complement. We'll receive the reverse complement of the DNA string. Oops. DNA seq, I'm sorry. And we're going to return print rev c. Okay, so once we define our self function, we're going to use if name equal to main. We're going to run solve. Okay, looks nice, right? So let me just prepare my download folders here. I'm gonna click it and cross my fingers. Okay, let me check it 1 second. Okay, so this is our file. Okay, is the revc5. So I'm gonna copy the relative path here. I'm gonna use it. Okay. So now I'm gonna back to the I'm gonna run Python. M now is source scripts dot revc. Looks nice, right? So let's copy it and back here and paste and cross your fingers, and the site's really slow today. Okay, we made it. Okay, so as we guys can see here, our problem are based on the definitions of, I don't know why I do this, in the definitions of what is a DNA string, and we have to remember that the DNA molecule has two complementary strings. Okay, so if you guys like this video, I will ask you to give a like and subscribe to our channel. I hope you join me in the next videos, and I see you there. Bye bye.